This series is about biochemistry and structural biology of nucleic acids, and it's from a course that I gave for postgraduate students in Warsaw, Poland. It is structured into several different parts. I start out from the question why nature uses the building blocks that it uses in nucleic acids. I then cover classical themes of nucleic acid structure. From there, I move on to the biochemistry of DNA modifications, either from damage or from modifications that are actually enzymatically catalyzed. In this part, you will hear about why nature uses the building blocks that it uses. Take inspiration from Eschenmoser in Zurich, who in turn quotes Einstein himself, who says, we not only want to know how nature is and how her transactions are carried through, but we also want to reach, if possible, a goal which may seem utopian and presumptuous, namely why nature is such and not otherwise. And for a very long time, of course, this was a completely impossible question to ask. But basically, since Eschenmoser's landmark paper in Science in 1999, actually, it's become pretty clear for nucleic acids why nature is such and not otherwise. And the aim of this lecture is to share a little bit of this understanding also from very recent work that I found very instructive and informative. So the basic presumption is that what you want as an information storage polymer is some repetitive polymer that looks uniform from the one side and sort of from the other side has a variability that encodes a sequence of letters in the way that text encodes a sequence of letters. And of course, in an original RNA world, or then in the modern biological world with DNA as the information carrier, you have the backbone with the phosphates and either riboses or 2' deoxyriboses, and then the bases are based on purines and pyrimidines. But of course, these are a lot of special choices. And I think for most people, it's not really clear why nature would have made these choices as opposed to other ones. So Eschenmoser sees this more sort of as a chemistry question. If you hear someone like Benner from Florida, previously also from ETH, then he says, if there was life on some exoplanet somewhere far out in the universe, what would you expect it to look like? And basically, this is not just a speculative question, but it can be astonishingly well answered chemically. And let's first look, let's assume that we have a linker, the phosphates, that we have an anchor, the ribose or the deoxyribose, and the letters, so the purines and pyrimidines, and let's understand for all of them why nature chose what it chose. So let's start with why phosphates. And actually, there's a very classic answer to that in a famous review article in Science by one of the really leading physical chemists of his day, by Westheimer, who basically says, look, phosphates are very widely used in biology. And on the other hand, chemists actually strictly avoid phosphates. So what's so good for phosphates chemically? This is not the subject of this talk, but it's a very interesting discussion by Westheimer. And what is so good in phosphates for the linker unit of nucleic acids? And basically, Westheimer's conclusion is that it can make connections two ways, so it forms the diesters, and it then still has a resulting negative charge that stabilizes the diesters against hydrolysis and also helps
to retain the molecules within a lipid membrane. So it's localization, and more importantly, it's long-term stability of the linkages against hydrolysis. And trick to assure the long-term stability is the negative charge. And the argument that he makes for that is to say, okay, the negative charge attracts protons in the same way that it repels negatively charged nucleophiles by the same amount, but of course with opposite sign for unit charges. So by looking at pKa values, which measure the proton affinity, you can learn something about how much the extra charge affects the next step. So how much an extra negative charge makes it harder to remove a proton, makes it harder to remove a proton, or makes it harder for a nucleophile to approach. And basically, Westheimer's comparison is citric acid versus phosphoric acid. And what you see is that basically the pKa's or the K values, so the equilibrium values, tell you that the negative charge has a huge effect. So basically, the negative charge is the guardian angel of the phosphodiester backbone stability. Now, there would be various ways to have negative charge, and the obvious sort of counter alternative for phosphates are the arsenates, right? And the arsenates are simply too unstable. And the same applies to the to the silicates, and there you additionally have the problem that they're too basic, so that you don't actually have the negative charge. What is less easy to see that it's not good is what if you took something like citric acid on the left? And the answer is that for the negative charge really to protect against nucleophilic substitution, it has to be really close by. So in citric acid, the negative charge that is left after you form two carboxylic acid diesters, basically negative charge is so far away that it affords almost no protection. Remember the previous pKa table. So what we learn from this, that actually with respect to the linker unit, actually nature's choice is really wise to assure long-term stability. And with that comes a funny anecdote that I still remember when it was published. So about 10 years ago, there was a paper in Nature, I think it was Nature, that actually claimed that there's a bacterium from a lake in California that's very salty and also full of other exotic elements. And the conclusion was that basically this bacterium tolerates insane amounts of arsenic. And then probably to get it published in Nature, the authors insinuated that the arsenate may actually be partly or fully a replacement for the phosphates in the backbone. And of course, someone like Westheimer would have known decades before that this has to be nonsense because of the stability. And sure enough, the paper collapsed very quickly. So in fact, even an organism that lives really in very exotic conditions still chooses to work with phosphates only because they're so good for them. Now, we've covered why the linkers are phosphates. So why would we have deoxyriboses? And basically, this was really single-handedly Eschenmoser's work initially to turn this from a lot of hot air speculation in the field to real synthetic organic chemistry. And his research program was basically to say, let's model DNA or RNA-like polymers with other anchor regions, other sugars, and see which one would at all be plausible. And let's then synthesize these and see how they behave. And this is the program that is laid out in a science overview that I really recommend very, very highly. Absolutely classy work. So, of course, assuming you still think inspired by the riboses, you think, what other rings could you use? And then, of course, it's pretty quickly clear that you want to play with 5-membered rings and 6-membered rings, because, of course, 4-membered rings are incredibly strained and unstable. So Eschenmoser assumes you want 5-membered or 6-membered rings, and he additionally assumes that the scaffold should be sugar-based. So the formula should be CH2O and then multiplied by the number of carbons. And basically later on, some deoxy substitutions are possible, but this is the basic concept that he wants to explore. And what he actually does is, so he says, okay, let's take hexoses, and he does a lot of modeling, and he then basically decides that on the computer, at least certain alternatives to the ribose might be possible. And he, of course, sticks to the D series only, as in the natural nucleotides. There's no point in repeating the mirror symmetric experiment. And actually, results are very, very interesting. So it comes out that the strength of base pairing is actually dependent on the backbone and is dependent on the sugar that you choose for the backbone. And for quite a few ribose or deoxyribose alternatives, what he actually saw is that he had inferior base pairing strength and much worse selectivity compared to natural DNA and RNA. So one usually thinks of base pairing as a property of the nucleobases and not of the backbone. But that's actually a partly mistaken concept. And it actually also very much depends how you present the nucleobases. And apparently, these alternative scaffolds are bad at it. Some are even so bad that by steric hindrance, they actually altogether hinder the formation of proper duplex formation. So a lot of the sugars, and specifically of the hexoses that Eschenmoser was actually testing, are clearly no good at all. And then perhaps the biggest surprise of all, when he used dideoxyallose as the anchor for his bases. What he basically found is that even though he uses natural nucleobases, now suddenly you don't get natural Watson-Crick pairing anymore, but you get exotic other base pairings. Like you have GC is still best, as you would expect, but then the next best base pair is AA or GG, followed by AT. So actually, we always think that base complementarity and Chargaff and Watson-Crick pairing are nucleobase properties, but actually it depends on the backbone as well. Depends that you present bases in the right way. That to me was a big surprise. And then an even bigger surprise perhaps comes when he played with pyranosyl ribose. And what he now sees is that suddenly he does get a duplex that's actually even better than natural dsRNA or DNA with respect to melting point. So it forms tighter duplexes. And actually you also exclusively get Watson-Crick base pairing. So basically the conclusion with respect to the anchor from all of this work is that there are lots of ways to mess it up in rather subtle ways. Lots of things can go wrong that at least I would not have had on the radar at all, that they could go wrong. I would never have expected base pairing to go wrong if you play with the backbone. And surprisingly also, that there are solutions that, at least when base pairing specificity according to Watson-Crick and duplex stability are your goal, right? Nature's solution is not actually necessarily the very best one in this respect. So the question then, of course, is why would nature still choose the riboses and deoxyriboses? And one of the possible answers is that there are good prebiotic routes how you could make these building blocks. And actually, I don't want to go in detail into the chemistry, but have a look at this really interesting paper from John Sutherland's group, which basically shows that it's possible to, under abiotic conditions and without enzyme catalysis or any sophisticated means, to basically stereoselectively synthesize the building blocks under prebiotic conditions. And of course, this is a bit of a fallacious argument. This was a big research program in the UK. I remember lots of talks on that in Cardiff. And of course, everybody was trying to find chemical routes to the chirally pure nucleosides that nature is actually using. Not nearly as much effort has gone into attempting to make the non-natural alternatives. So it's not sure whether people wouldn't come up with good ways for that as well, but it could be one argument that favors the choice of the deoxyriboses. So when you cast the net wider and go away from the idea that you want to have these regular sugars, then in fact you can have a lot of alternative anchors. And as you can see, arabinose has been used, a locked ribose has been used, fluoro-substituted compounds have been used. So there's by now a whole zoo of different anchors that actually all work. And remarkably, quite a few of them are actually capable of repeating information from one strand to the other. So there's still base pairing, and astonishingly for quite a few of them, there are also no variants of a replicative polymerase or of a transcription enzyme that basically allow to do synthetic chemistry with these non-standard DNAs or RNAs. So really, really impressive. And you can have a look at the science paper from the Holliger group. That is, to my knowledge, the first real breakthrough in this direction. So there are also solutions that are kind of more primitive. You can use peptide bonds instead of phosphate esters, and then you have the peptide nucleic acids, or not shown here, you could use morpholinos. So these achiral backbones actually will nicely give you hybrids with natural nucleic acids, but of course you have trouble if you want to use them on their own in the absence of a chiral nucleic acid. Why purines and pyrimidines? Right? So we've seen why phosphates. We've kind of seen with lots of caveats why riboses and deoxyriboses are good. We've also seen that they're not the only possible choice. Why purines and pyrimidines? And one of the classic answers to that question is actually that they are relatively well defined with respect to their tautomeric state. So for that, I need to explain a bit what tautomeric state means. You can see it from this illustration from Watson's book, The Double Helix. So basically, if you have a, say, in guanine, a CONH group, right? You could have the proton either on the oxygen or you could have it on the nitrogen. So look at the bottom diagram. And basically, was not clear in Watson's time, and it was actually a big obstacle to the discovery of the structure of double-stranded DNA. But it's very clear now that what's called the keto form, so the hydrogen on the N1 of guanine and not on the O6 of guanine, is actually very strongly preferred, right? Think amides, and you will actually expect this outcome. And this is important because if you mess up the tautomers, if you have equilibria, the coding properties of the nucleic acids change. So many alternative systems of nucleobases that could be used instead of the nature ones would actually be poor coders because they have equilibria that are more sort of on the side of equal population of two states with different hydrogen bonding patterns. And that's, of course, a killer for any information coding polymer. So sure enough, what you want is fixed preferences for tautomers. And then the other thing, and this is actually very nicely described again in the double helix, where Watson explains what was the reasoning that led them to their dsDNA model. And actually his key point here is that the shape of a guanine cytosine pair and the shape of an adenine thymine pair are actually almost identical. And he describes his joy when he realized that this actually means that you can make a very regular polymer that is variable on the inside, but presents a sort of uniform outside, which is important for wrapping on histones, say. So the uniformity of shape of all the possible base pairs is a key criterion for an information containing polymer, and clearly the purines and pyrimidines fit. And that's actually Watson's big insight. It's illustrated here. So you see that the shape of an AT pair and of a GC pair are really very, very similar. Note the position, I don't know whether you see my mouse, of the C1 kind of the ribose or deoxyribose that is basically in the same place everywhere. And also note that if you swap the A and T or you swap the G and C, still overall, you still have pretty much the same shape. So all four base pairs actually have roughly the same shape. And that is very, very useful to make regular polymers. And it actually was not true initially for many of the synthetic alternatives that were developed. So this is an example in Jacques, actually not so long ago, of what they called fat and skinny DNA. And actually there basically you violate the principle. But there are meanwhile also really nice systems that again show that even based on the purines and pyrimidines, you could go further. So there's something called the Hachimoji DNA, and apparently Hachimoji, which I probably pronounce awfully, means 8 letters. So it's actually possible to make DNA that has 8 different letters rather than just the 4 standard ones. And the group that made this, and that's a science paper from only by now 4 years ago, I guess, that they even made a polymerase that can synthesize they made a T7 polymerase that can synthesize 8-letter RNA from the 8-letter DNA, quote in quotes. And actually, in terms of coding capacity, this would allow you to go down to a 2, not to base triplets for the code, but base doublets would do, because you have 8 alternatives, and then 2 of them would give you 64 possible combinations, which is just as much as you have from 3 different standard DNA or RNA bases in the triplets. But of course, you would pay a fidelity price, because some of the pairs differ only in a single hydrogen bond. So you could store information much more compactly, but you would pay a price in the fidelity of the information that you store. So it's still that, at least on basic biophysical grounds, you expect that the natural DNA is superior